let's talk about our last group of macromolecules, nucleic acids. Nucleotides, which are going to be making nucleic acids, it's going to be mainly contained, or nucleic acids are mainly going to be composed of, again, carbon, hydrogen, oxygen, nitrogen, and phosphorus. Okay, these are going to be like the main elements to make nucleic acids. Now, nucleic acids are going to be our polymers, okay, our chains. And what is going to make a polymer? Monomers. The monomers of nucleic acids are going to be called nucleotides. So a nucleotide is a monomer. You put a bunch of monomers together, you make a polymer. So you put a bunch of nucleotides together, you make a nucleic acid. The main roles of nucleic acid or nucleic acids. The main roles are going to be storing genetic information. Okay, important. Storing genetic information. They're going to provide instructions to build what? To build proteins. And we spoke about in my previous video about proteins, right? Proteins are very important. They make you. Okay, they're for structural purposes. They do it for enzymes, but that's what's shown, right? That's what makes you really. But where are the instructions to make those proteins? We talked about it. It's going to be in a molecule called DNA. Now, there's two types of nucleic acids. Okay, two types of nucleic acids. We have DNA and we have RNA. And really, one of the changes between these two, it's the type of sugar that they'll have. Okay, DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. And I'll show you the sugar in a little bit to show you the difference between both of those. Nucleic acids are going to be long polymers of nucleotides being the monomers. Okay, and if you can see here in this picture, this is a little bit of an example of what we're going to be seeing in a little bit. Okay, a nucleotide is going to be this one over here, this part over here. This is going to be a nucleotide. Okay, we'll go into more detail in another slide. Here is going to be another nucleotide. Etc., etc., etc. You see, and how do we bring nucleotides together? Again, it's going to have to do some kind of dehydration reactions. Okay. Okay, this is what I was explaining to you in the previous video, and let me expand a little bit more. Okay, nucleic acids, why are they so important? Because they're going to be storing and transmitting hereditary information. DNA, as you see over here, DNA, and we'll talk about this in a different chapter, or we'll expand on it. Okay, DNA is the genetic material inherited from our parents, right? Our blueprint. And they're going to be huge macromolecules. Okay, I want to call this the blueprint. Well, DNA, okay, is going to be made of a bunch of things called genes. Okay, so a DNA strand can have lots of different types of genes. Okay, which leads us to the question: what is a gene? A gene is a unit of inheritance encoded in a specific stretch of DNA that programs the amino acid sequence of a polypeptide. So a gene is going to tell us, it's going to code for a protein. Okay, a gene codes for a protein. Now, how does this work? This is a very simplified picture. Everything starts with a gene, okay, with the DNA. A gene is nothing but a piece of DNA. There we go. Just to make it very simple and you understand this concept, okay? A gene is a piece of DNA, just this piece from here to here, which is going to code for a protein, okay? From there, what's going to happen? We're going to go through a process called transcription. And please write this down because this is going to be something very basic, but we'll expand this. We'll talk about this several times, okay? As we go in this chapter, then in the cell chapter, we'll go through this again. In transcription, what we're doing is we're making a copy of an RNA molecule from a DNA molecule. So we say we transcribe DNA into RNA. Once we have RNA, the RNA can exit the nucleus of a cell. It goes, it finds a ribosome, and the ribosome is going to read the mRNA and make it into a protein. And that process is called translation. Why translation? Well, think about this, right? If there's someone who doesn't speak Spanish, so, and someone else is speaking English. So what happens? We have to translate. And the reason why we have to translate is because we have to change the language here. We're going to go from a DNA language, which is nucleic acid, okay, into another language, which is amino acid. So that's why, that's what, think about that when you think about translation. What is translation? We're making RNA into proteins, but the reality is we are reading this nucleic acid language and we're making it into a different language called a protein language, into amino acids. So I want you guys to understand this concept here. From DNA to RNA, we go through transcription. Okay, and from transcription, now we have our mRNA molecule. Then a ribosome is going to be able to read this mRNA molecule via a process called translation to make a protein. And again, we'll expand on this a little later on in a different chapter. Okay, let's look at the nucleotide really quick. Okay, let's look at the nucleotide, the monomer of a nucleic acid. The nucleotide is a monomer. A nucleotide is composed of three main things. Okay, one, two, and three. Okay, this is going to be a nucleotide. Okay, that's a nucleotide. Three main things. One, a 5-carbon sugar. Okay, and this is going to be our 5-carbon sugar. A phosphate group. This is going to be a phosphate group. Okay, made of phosphate and oxygens. Okay, and finally, a nitrogen containing base. I usually call it a nitrogenous base. Why nitrogenous base? Because he has nitrogen in it. It's composed of a lot of nitrogen, and this would be the nitrogenous base. Okay, are they all going to be the same? No, we'll explain that in a second. There's going to be 4 different types of nucleotides. Okay, but I will explain that in a second. You see how the nitrogen containing base contains, guess what? A bunch of nitrogen. You see, it has a lot of nitrogen in it. Okay, that's why it's called a nitrogenous base. Okay, a couple of things. The phosphate group doesn't really change with any of the nucleotides. Okay? So that's going to stay very much the same. Okay, this phosphate group, it's going to give a little bit of a charge. It's going to be able to link to the next chain. Okay, but besides that, let's look really quick at the sugar. Why do I want to look at the sugar? I'm going to change this color because the sugar, okay, this nucleotide, how do we know if this nucleotide is part of it's part of DNA or RNA? The sugar in DNA is going to be called deoxyribose. Okay, that's deoxyribose is the sugar in DNA. What is the sugar in RNA? The sugar in RNA is ribose. And what is the big change? What is the big difference? If you look at here, look at the sugar. Actually, there's the labeling there already. I don't want to over label things. What is the difference between ribose and deoxyribose? Well, if it's deoxyribose, these are, okay, is to be a hydrogen. So if this were a DNA sugar, this would be a hydrogen. Simple as that. Okay, deoxyribose, the sugar is going to have an OH and an H. Okay, a hydrogen. That is a DNA sugar, a deoxyribose. Now let's make this into an RNA sugar. I'm going to erase it really quick. Okay, and I'm going to use a different color. I'm going to use green. How do we make this into ribose? Same sugar, except that in here, instead of having an H, we're going to have another OH. Okay, so one OH here, one OH here. Now we made ribose. That's the only difference between the sugars, between a ribose sugar and a deoxyribose sugar. Okay, so far so good. All right, that's one thing. Another thing I want to make, another point I want to make is, let's talk about nitrogenous bases. Okay, nitrogenous bases. In terms of DNA, you have four types of nitrogenous bases. You're going to have adenine, guanine, cytosine, and thymine, T for T. But when we talk about RNA, RNA is also going to have adenine, guanine, cytosine, but instead of thymine, RNA is going to have something called uracil. We'll expand that in a little bit. I just want to give you an idea, okay? Just want to give you an idea. One more thing I just want to point out of this diagram is this. I'm going to erase this, so please feel free to pause it, pause your video, and put your notes. DNA and RNA are said to have a sugar phosphate backbone. So the backbone of the nucleic acid is going to be called a sugar phosphate backbone. Why? Because this is going to be the backbone of your DNA. That's going to be the backbone, okay? Sugar phosphate backbone, okay? It has to do with the phosphate part and the sugar part of the nucleotide, okay? And you will see how they're going to be linking with each other. What about the nitrogenous base? The nitrogenous base is going to be floating around. It's going to be, think of it as teeth. Okay, it's going to be sticking out as teeth. Okay, I'll show you information one more time in a later section, in a later slide. Okay, so things to know about this slide. Again, a nucleotide is a monomer. Okay, you have a lot of monomers together, you make a put a bunch of monomers together, you make a nucleic acid. A monomer or a nucleotide is going to have a 5-carbon sugar. Okay, if it's DNA, it's going to have deoxyribose. If it's ribose, it's going to be RNA, depending on the sugar, and I show you the differences. A phosphate group, the nucleotides have a phosphate group, and all monomers are going to have a nitrogen containing base, a nitrogenous base, which could be adenine, guanine, cytosine, or thymine if we're talking about DNA. If we're talking about RNA, nitrogenous bases, we're going to have adenine, guanine, cytosine, but instead of thymine, we're going to have something called uracil. Okay? Nucleotides, let's get a bunch of monomers, a bunch of nucleotides together and make a nucleic acid, right? A polymer. How do we do that? Same thing as we've been doing over and over and over and over, right? So nucleotides are going to be linked by a phosphodiester linkage. Let me go back to my red. Nucleotides are linked by phosphodiester linkages. Occurs by what? If you guessed dehydration reaction, you're absolutely right. Remember, when we're building up big molecules, we have to go through dehydration reactions. It's going to link the 5' phosphate to a 3' hydroxyl group. Okay, and the nucleic acids are going to have a 5' end, phosphate end at the end, and a 3' end at the okay, at the hydroxyl group. Okay, so let's put them together. Let's look how this reaction occurs. Okay, this is one. this is proteins, by the way. Just giving you an example of what happens in proteins, and we talked about this. So this is just to kind of flow you into nucleotides and how they get together. Okay, um, two amino acids. This is one amino acid. Okay, another amino acid. You have a dehydration reaction, sometimes called condensation, which means we lose water. We lose water, and we make a peptide bond, and now we have a chain of two amino acids. Well, now let's look at the synthesis of a nucleic acid, and the same thing. What's going to happen here is this hydrogen from here and this OH from this phosphate group is going to go away, right? We're going to get rid of it. We're going to rid of it via dehydration reactions or condensation reactions. And now we brought two together, okay? And this is going to be our phosphodiester linkage. Okay, this is going to be our covalent bond. This is how we get one nucleotide together with another nucleotide. Okay, and we always add it at the we always add a new nucleotide at the three prime end of a molecule. Okay, and that would be the first part of our nucleic acid section.